Hi, Year 12s. Welcome back to our last part of the proteins topic. So again, we're diving back into our learning intentions, which are the last kind of parts. You'll be able to explain the role of proteins as a nutrient in our diet and understand the biological value, or the BV, of proteins, including complete and incomplete. And again, don't forget, these are our words that we need to remember, understand, so adding them to our glossary is a must. We'll be covering function and the role of proteins in this slide. Whether essential and gained via consumption, or non-essential and synthesized by our bodies. They are linked together in a variety of sequences to make different proteins required for our body. The peptide bonds, which link each of our individual amino acids, so remember there's 20 of them, they link together by these peptide bonds, forming what's called a polypeptide chain. So this long chain connected by these polypeptide links. This is completed through a process called condensation reaction, which is the same type of reaction that combines our monosaccharides to form a disaccharide, or a disaccharide to a polysaccharide, and also in the same process of creating our fatty acids and glycerol chains to form our triglycerides. So the good thing about our bodies is once something works, it tends to stick with it. And when we look at a lot of these macronutrients, we're understanding that they're built from smaller units into larger units through a process called condensation reaction. So if you are in chemistry, you'll understand a bit more about the process, but condensation is the combination of two molecules together to form one larger molecule by producing water as its byproduct. And as we can see here, this is an example of a polypeptide. If we zoomed right into this section, we would see that there's a phenylalanine, and then there's a leucine, and I think it's a serine and a cytosine. All of these link together by these polypeptide bonds to create this long peptide chain called a polypeptide. And eventually this polypeptide, once made in our bodies, bends and folds down into a three-dimensional structure to create the protein with their specific function. So why are these long polypeptides really important for our bodies? So proteins range from a few dozen to several hundred amino acids in length. So these polypeptides can be just a few dozen or all the way into 100 amino acid chains. Their unique shape is determined by the amino acid sequence that is involved, so which amino acids are in each order. And this enables a different protein to be folded and different functions in our body. The sequence is dictated by a gene order, which is used using different cell combinations to build specific proteins for specific specialized roles. So if you are in Year 12 biology, you'll understand about the process of creating proteins through translation. This is that process, and the ability to fold into different proteins for different functions. Don't forget that a gene is a large segment of DNA, and that DNA dictates what of these amino acids will be placed where, and how that protein will fold, and how that protein will function. So histone proteins are an example of a protein used in our body. These proteins play a structural role in cells and is important for the condensation of our chromosomes. So the compaction of our chromosomes by having our DNA wrap around them and then compact further into our chromosomes. So chromosomes consist of double helix DNA molecules wrapped around what's called a little histone. So these kind of like little circular discs. They're wrapped around almost like a spindle or spool that you would see different types of wool maybe wrapped around, and they contain these linker positions as well. Eventually, they're kind of folded on each other, further compacted down into our chromosomes. This provides stability, protection, and also helps regulate our protein production. So only the ones that are open are able to be accessed. If it's really tight and compacted, it's a way of stopping these genes and proteins from being produced. So why is it important? Well, our proteins are involved in growing, maintaining, and repairing our body tissues. They're essential for growth, maintenance, repair. And when consumed, proteins are broken down into our amino acids. So we talked about how there are 9 essential amino acids, and there are a total of 20 amino acids, and 11 are classed as non-essential. Our cells need these 9 essential and also these non-essential to create these protein structures called our polypeptides. So we need to make sure we have enough of these proteins present in our diet, or the ability to break apart proteins and create more proteins through synthesis. From conception, proteins are needed for development of our bones, muscles, nerves, and more. And the body replaces around 330 billion cells per day, which requires our protein to repair our tissues like our skin that's cut, bones that are broken, and also potentially our fatigued muscles. Proteins have a role in synthesizing things called enzymes, which are proteins, and synthesizing means to make, to build, to create. So an enzyme is a catalyst which speeds up the biochemical reaction, such as digestion, tissue building, or converting glucose to energy. These are all processes that require proteins that help us make it go quicker, called enzymes. Each enzyme has a specific shape, and this is called the active site, which attracts what's called a complementary substrate. So the substrate is the thing that is being digested, being built, or being converted. Okay, so this substrate goes into the active site. Hopefully it's the right shape or conformation. It sits within, and there is a chemical reaction that's undergoes in this active site where we can either build or break. So this is an example of a catabolic enzyme which breaks apart into 2 proteins, or we could have ones that build something up into larger, which is called anabolic. We've got this example here as well, again another catabolic enzyme reaction where we have maltose as a part of our disaccharide. Enzyme comes in, chops in the middle as it's fitting into our active site. It's cleaved, and 2 molecules of glucose are released through this process, which is a catabolic enzyme, larger going to smaller. Don't forget topic 1. Please complete all topic 1 questions from page 75.